The formation of proteins from tRNA is a complex process. Basically, it starts out with a tRNA molecule, which has an amino acid attachment site and an anticodon, which is a nucleotide triplet, which corresponds to a specific amino acid. And then this tRNA enters the ribosome into the A site, then it goes to the P site and the E site, which correspond respectively to the codon recognition, the peptide bond formation, and the exit or translocation. The tRNAs can bind to more than one codon because the last base of the codon is flexible, which prevents major disruptions in protein function if one nucleotide is off.
Amino acids. There are 20 amino acids that form proteins. There are more, but those are uncommon and not found typically in most proteins. You can classify amino acids into 6 different structures, or in 6 different types. I'm not going to read them out because it's not really essential to know, but the basic structure of an amino acid is an R group, a central carbon, a hydrogen, a carbonyl group, and an amine group. Here's a picture of the amino acids. There are 4 basic protein structures: primary, secondary, tertiary, and quaternary. Primary is essentially a chain of amino acids that forms a polypeptide with no folding. Secondary includes alpha helices and beta sheets. Here is a picture of an alpha helix and a beta sheet. This is an alpha helix and this is a beta sheet. That's secondary structure. Tertiary structure is the overall shape of a protein, but only from one polypeptide. So there are 3 main forces that shape the tertiary structure of a protein: hydrophobic interactions, polar side chain and ionic bond interactions, and disulfide bridges. Finally, quaternary structure is pretty much what is found on most proteins, as most proteins are made with more than one polypeptide chain. Essentially, it's the same as a tertiary structure, except that it includes more than one strand of a polypeptide. So, and what basic
There are several different functions of proteins. One is a contractile and motor function, in which proteins enable the movement or contraction of muscles or cells. The other is the signal function, in which proteins act as hormones or receptors of hormones to convey a message within the human body. The other is transport, in which proteins move polar molecules into the cell or any molecule against its concentration gradient in diffusion or active transport. The third is structure, in which proteins provide structures. These proteins are usually long and fibrous. The next one is storage, and these proteins store amino acids for the body's use. They're also found in plants. Another one is antibodies, and for the immune system. Antibodies are proteins, and they are used in identifying and neutralizing potential pathogens. The last one is the enzyme function, which will be discussed in the next section. Alright, so most enzymes are proteins, and what enzymes do is they catalyze a reaction from a reaction. So if a reaction is considered to be a trip over a hill, then the enzyme is the engineer that digs a tunnel through the hill to enable an easier path for the reaction to take place, which is analogous to the lowering of activation energy that a catalyst enables. So most enzymes have a substrate, and the substrate is basically the compound or compounds that fit into the active site of the enzyme that then react within the active site and then releases products. And these enzymes and substrates fit as key and locks. But it's not an exact fit until the substrate enters the enzyme, and the reactions between the enzyme and the substrate cause induced fit. Most enzymes can be inhibited by competitive inhibition or non-competitive inhibition, in which either one substrate mimics another and takes its place in the active site, or another molecule binds to a different site of the enzyme and changes the shape of the enzyme, which causes the other substrate to not be able to bind to it.
Protein degradation is carried out through 2 pathways: ubiquitin proteasomes and lysosomal proteolysis. Ubiquitin is a marker molecule that attaches to the lysine residue of a protein, and once it forms a long enough chain, it'll be recognized by proteasomes, and it'll be degraded for the ubiquitin to be reused. Lysosomal proteolysis is essentially where lysosomes take up proteins and degrade them. They can take up both proteins from within the cytosol and from proteins outside of the cell through autophagy, which is a mechanism to engulf proteins.